Welcome to Plastisphere, the podcast on plastics, people, and the planet. My name is Anja Krieger. When I started this podcast 6 years ago, a new field of research had just emerged, the science on nanoplastics. Nanoplastics are pieces of plastic so small that you cannot see them with your naked eye, even smaller than microplastics. Back in 2018, I visited Alexandra Terhal in her lab at Paul Sabatier University in Toulouse.
Bonjour Alexandra, ça va?
The French chemist and her team were the first to detect nanoplastics in the open ocean. Can you compare it to anything in nature?
Viruses. A nanoplastic is just big like a virus.
Tiny, like a virus, yeah.
Yes.
And if you're looking at the risks, what is it you're looking at?
Well, we are wondering if they can be transferred in the body. We are looking if there are some consequences in the organisms, genotoxic effects or mortality or change in the behavior of the animals. We are looking at all those kinds of things.
The science had just begun and there were 1,000 open questions. But in the past 6 years, a lot of new research on nanoplastics has come out. And recently, there's also been more and more reporting in the media, citing pretty scary findings, that nanoplastics have been found in blood and all kinds of organs, for example. But what does the science really know for sure at this point? I connected with one of the leading scientists in the nanoplastic field, Denise Mitrano. Denise is a professor at the Department of Environmental System Science at ETH Zurich, the Federal Institute of Technology Zurich in Switzerland. Hi Denise, thanks for joining us on the podcast.
Hi Anja, thanks for having me.
If you made a map of the knowns and unknowns of nanoplastics, what would it look like?
Well, unfortunately, the known parts of the nanoplastic story of this map need to be much more explored than what is still unknown or hypothesized. So we do know that nanoplastics exist in the environment. We see macroplastic waste that is in the environment, and we know that through UV and mechanical stress, this can break down into microplastics, those particles that are 5 millimeters in size and below. And consequently, this fragmentation can continue to the nano size. So nanoplastics are likely to be found in every environment that plastics and microplastics are, even if they're not yet detected. So we know that they're there. The big questions are, how do they get there? How do they move around? And what are their impacts?
Especially in the past few months, there's been a lot of news reports in the media that sounded quite concerning. So let's do a little media review. I'll read you a couple of quotes, and you, from the scientist's perspective, tell me what you can say about each one, okay?
Okay, great. Let's get started.
So this first one is from May 2024, so just a few months old, from an outlet called The Conversation. Nanoplastics are attracting growing concern thanks to recent technological advances that have made researchers more able to detect and analyze them. Their smaller size means that they are more easily transported over long distances and into more diverse environments than microplastics. They can more easily penetrate cells and tissues in living organisms, which could lead to different and more acute toxicological effects.
Yeah, so with this quote, I think that it's not necessarily surprising that we find nanoplastics in many different environmental and biological media, because the same has been shown for microplastics as well, maybe not on the cell level, but in many places. So that nanoplastics exist in different places is not necessarily surprising. The most interesting and intriguing part that we still need to find out is the so what. So we find nanoplastics there, and what is the actual issue? This size continuum of plastic pollution, though, is very important because smaller size particles generally can be transported further, and they can also have more ability to pass through biological barriers or even have cellular interactions. So this size continuum of plastics pollution is really what makes nanoplastics a specific concern compared to the other size classes of plastics pollution.
Right. So we have the potential, but we don't have the evidence that it's causing huge impacts, right?
So there have been some suggestions that impacts are happening, but as with all size classes of plastics pollution, we still are not always sure if these impacts are from the plastic particles themselves or the plastic added chemicals and metals that are put into plastics for different purposes. And so we still need to also tease out what is actually causing the hazard. Is it the presence of a particle? Is it the type of that particle? Or is it what is contained and leached out of that particle?
And is that something that we can find out in every case? I imagine that might be really hard given like the many variables, the many kinds of chemicals that can be in one plastic and also the environmental conditions that can vary.
Indeed, it'd be a never-ending list to be able to test all different types of plastics with all different types of plastic-added chemicals, as well as putting them into so many different environmental and biological contexts. So I think this really also comes down to the study design and what is the targeted question that the researchers really want to investigate. So it is possible to decipher some of these differences, but it depends on what is the intent of the study design.
Mm-hmm. Okay, let's hear the next quote. This is from March 2024, and it was published on CNN. Nanoplastics are the most worrisome plastics for human health, experts say. And then the article explains that nanoplastics could migrate through the digestive tract or lungs into the bloodstream and invade cells and organs, as we heard. But it also says, until now, however, research has yet to determine just what impact those polymers may have on the body's organs and functions.
Yeah, I think that this quote really shows some balance from the media outlet, because while it does say that nanoplastics are the most worrisome plastics, and as we discussed in the previous quote, maybe there is still some room for debate there, they do acknowledge that there is still more research that needs to be done, since the actual mode of action of what is happening with these nanoplastics in humans or in organisms is not 100% defined.
Mm-hmm. Would you be one of the experts to sign that claim, nanoplastics are the most worrisome plastics for human health?
I would not necessarily be one of the scientists that sign this claim. This isn't to say that I'm not worried about nanoplastics in human health, but it's hard to say what is most worrisome. Again, this is aggregating a very large amount of information on a very diverse and heterogeneous group of materials into one small statement. As we already discussed before, there's a lot of different types of polymers, plastic-added chemicals, and metals that are in plastics. And so it's also important to remember that not all nanoplastics are the same. This is a wide variety of materials, which each may have different hazards.
So what are the differences between plastics that would make them different in terms of their health impact?
Well, this is still something that's being researched to actually link plastic properties with specific outcomes. But we do know that this heterogeneous group of materials contains a lot of different types of polymers. So PET, polystyrene, polyethylene, for example. And each different type of plastic, when it's made for a specific type of purpose, will also have different types of plastic added chemicals, which can change their color, make them more or less flexible, make them more or less brittle. And these different chemicals may then leach out of plastics at different rates, depending on the polymer structure, as well as the environmental or biological media that they're in. So indeed, it is an endless list of variables that you can mix and match across a large experimental matrix. And so that's why it's not unreasonable to try to group the different plastics into one umbrella term. But I think as a scientist, it starts to get a little bit harder to digest, no pun intended, when there are these big overlapping terms, when we know that there's actually a lot of variability within this space of one material called nanoplastics.
Right. I've got another quote for you from May this year on phys.org. Nanoplastics can accumulate in developing hearts, according to a study. And this article then goes on to explain that the study was done on chicken embryos. They say you can find nanoplastics everywhere now, in the sea, in the soil, in the food chain, and in human blood. And they also say that nanoplastics have been found in human placentas. And we found that the nanoplastics can cross blood vessel walls and that they accumulated to relatively high levels in the heart, liver, and kidneys. What can you tell us about that?
I think that this seems like it's very plausible from some of the research that we've also done in our lab or with collaborators. So in our group, we're also looking into different avenues of translocation of nanoplastics into different organisms, including fish as well as mice that are exposed to either water, which is contaminated with microplastics and nanoplastics, or food. We also looked at the association of nanoplastics with human cell lines and saw that the nanoplastics were either associated with the cell surface or also could be, in some cases, inside the cells as well. So the first examples of these, with the fish and the mice, we've been able to show some translocation across biological barriers, in this case, the gut specifically, but we can also show that nanoplastics are going into cells. So I think that this aspect of the possibility of nanoplastics being able to move into different parts of an organism is well shown through both experimental laboratory studies as well as when there are patients that these samples are collected from.
CNN also reported that tiny plastic shards had been found in men. Human testicles contain microplastics and nanoplastics at levels 3 times higher than animal testes and human placentas, they say. This isn't the first time plastics have been found in reproductive tissue. And the article notes that animal studies have shown tiny plastics can impact sperm count and contribute to hormone and other disruptions in male sexual organs. That sounds pretty concerning, doesn't it?
Yeah, indeed. I think that we also see across the globe that there are declining birth rates and issues with fertility, and everybody's wondering what is the cause of this, and could it be plastics or plastic-added chemicals? In this particular study, the plastics were found in these different individuals, both in testicles and in sperm. As it was already pointed out, that there's a very limited number of men that this was found in. And what the article doesn't seem to report, or the media outlet doesn't seem to report, is if all men were actually tested to contain microplastics or nanoplastics. So in that aspect, we don't necessarily know how widespread of an issue this is. Or furthermore, if there are any environmental factors or lifestyle factors which are contributing to these elevated concentrations in these individuals.
Right, right. Do you think plastics are over-reported in that regard? That if there's news like this, it gets more widely reported, whereas other potential factors in the environment or around us are not?
Well, I don't think that it's over-reported because plastics pollution really is everywhere, including nanoplastics. And so it's really great and important that people are informed about what exposures and impacts there may have. What seems to be a little bit lacking, in my personal opinion, is this contextualization. So it doesn't mean that something else is necessarily worse than plastics, or it's okay if you are contaminated with plastics because you're exposed to other things. But it's always important to put this into context of the harm of one specific chemical or material compared to all of the other aggregated factors in your life that could also cause some issues. I think about this often in the context of the lifestyle factors that I try to have in myself to have a healthy lifestyle. I'm not drinking too many sugary drinks. I'm not eating fast food. These are known contributors to obesity and heart disease and things that are really controllable. What is very scary for many people is the exposure to plastics seems uncontrollable. And there is really where a lot of the people, I think, are picking up on how shocking this result might be.
And my feeling is also, or my impression is from reading the news, that there are also some dynamics in the way media works today that really contribute to this discrepancy between a simple statement and the contextualization that you just asked for. So, for example, what I see a lot is articles that are pretty balanced when you read them all the way down to the end, but the title is clickbait, basically. And as we know, many people just read the titles and the teasers, and then that gets stuck.
Yeah, exactly. I think that, you're the journalist and not me. So that's why I really appreciate this more in-depth discussion with you in particular. But I think that this clickbait issue is a real one. And even when journalists are trying to present something that's balanced, sometimes they're under time pressure in order to not be able to get all the facts potentially, or they're under a pressure of how to shrink the story to fit into a shorter amount of space. Oftentimes, when I'm discussing with journalists the aspects of risk assessment in terms of both hazard and exposure, or thinking about uncertainties, there's oftentimes not a lot of space for that in an article. And so it gets boiled down to more simplistic phrases and more overarching phrases, which may not necessarily be correct in all circumstances.
Right. And the thought, I think, behind that often is to lower the barriers of entry, right? To write in as simple a language as possible. But then some of these subjects are so complicated that they get changed when you simplify them. And another thing I'm not sure you're aware of is, or listeners are aware of, is that authors, journalists often don't get to choose their own titles. So, you pitch the story, you do the interviews, you write the story, and then you send it to your editor and the media outlet. And then it goes into other stages of this process. And yeah, it's oftentimes, in my experience, not on the author to decide what the title says, which can, sometimes you have to follow up on that and be like, hey, this title doesn't represent my article. But that's reality in the media, unfortunately. I mean, some outlets are super correct and they will show you the whole story until the end, but it's not the norm from my experience.
So I know that you're the one that's supposed to be asking questions here and not me, but how does that make you feel about this? Or do you have the leeway to, quote unquote, fight back to change what you think is the correct story? Or do you worry that you won't get a next assignment if you don't write something in a certain way?
Right. Yeah, I mean, that really depends on your personality, your relationship with your editor, the outlet you work with, how you can collaborate with them. But freelance journalism, for example, which, many people who do the contents nowadays are freelancers. Some are staff, but many are not. And that means that they're paid by time or by lines, right? So by length of article, by words, by letters sometimes, by seconds or minutes. And, how much time you put into that is your decision. And so if I decide to follow up on a title that is misleading, then of course that takes me time as the freelancer, and I have to pay for that myself. I usually chose to do that, right? But it takes time for all sides. And as you suggested, this can also, of course, make you more unpopular if you're always coming back after the story has been published and you're like, hey, this is not correct. Please go back and change that, because that will cost time and money.
I guess also, so I'm totally understanding about the time pressures, because also when I'm contacted by journalists, oftentimes it's like, my deadline is tonight or tomorrow, please call me back within the next 30 minutes. And it's like, okay, I should just drop everything right now. Yeah. And I often, of course, try my best to be able to do so. But what I've also found interesting about the choice of topics is more and more frequently journalists are contacting me for preprint studies. So those are not peer-reviewed studies yet, but they've been found on a preprint server, which shows all the information. It, of course, does not mean that that information is inherently incorrect, but it just hasn't gone through the entire scientific vetting process to actually say, this is what other scientists think about it. And I know that several of these very shocking headlines have been produced on, as of yet, unpublished data. So I think that that's also something to take with a grain of salt, that the story might also change from the science point of view through the peer review publication process, and that's not necessarily always taken into account.
Right, right. Now, after our critical media review, let's go into the science that you and your collaborators are doing right now. What are the questions you are looking into currently?
So in our group, we have three main pillars of the research. The first pillar is different analytical method development to be able to measure and characterize particles more accurately. Specifically, this is with microplastics and nanoplastics currently, but we look at a range of different techniques to be able to measure and quantify these particles. The second pillar of our research, which is the broadest, is the fate, transport, and biological interactions of particles. This is mostly in soil and water environments. Here we have a few different types of studies ranging from technical water systems, such as wastewater and drinking water treatment plants, to natural water systems, how particles are flowing down rivers or settling in natural water bodies, or even being incorporated into sea ice. When we think about the soil side of things, we look at transport through the soil profile, either with advective transport, so movement by water, or by bioturbation through worms and plants and things like this. We can then also, with collaborators, look at the ecotoxicity of different types of microplastics and nanoplastics, looking at things from cells to organisms to trophic transfer, so when one organism eats another. So there's really quite a lot going on in this space in the second pillar of our research. The third and final pillar is looking at policy, regulation, and material design. Because sometimes I find that as an environmental scientist, we're always just pointing out the problems and not being an active part of the solution. So I think that it's really great to be able to contribute towards science-based decision-making and policy-making, as well as working with companies to produce more sustainable and less negative, environmentally impactful products and materials.
Unfortunately, I can't be at your lab and see your experiments, but maybe you can tell us a little bit about your favorite experiments. You told me that you have some nifty setups. What's your favorite experimental setup that you've done or that you're doing?
So I would say it's not necessarily one specific setup that I find most exciting about the type of work that we do, but one specific tool that I find the most exciting. One of the main challenges of micro and nanoplastic research is actually the analytical chain, how to sample, extract, and quantify micro and nanoplastics in a given sample. Now, this challenge with analytics is the same in the lab as it is in the field, because you still have to be able to use oftentimes quite time-consuming techniques to enumerate and characterize your particles. So in our lab, we've developed an alternative approach, specifically in laboratory or pilot-based experiments, where we have metal-doped plastics. So here we include a trace metal into plastics when we produce them, and this metal can be used as a proxy for the plastics to look at their fate, transport, or biological uptake. The reason why we do this is we can use a trace and rare metal that is not already in the environmental system that we're studying, and then be able to use very fast and quantitative analytical tools to be able to measure this metal. And in that way, we can measure many more samples throughout our study, and we can start to better understand the processes and mechanisms of fate, transport, and biological uptake.
What is the hardest environmental medium to detect nanoplastics in? I mean, I know that it's in, oceans, lakes, rivers, so that's all water systems. It's also like blowing through the air, snow, soil. We've got many different places where plastics end up. So what is the hardest for nanoplastics research?
Well, for sure, each of these have their own challenges. So none of them can be considered easy. But when we think across the analytical chain, I think that soils are the most challenging. When we think about water, in contrast, we can filter a lot of water and pass it through a filter and collect micro or nanoplastics, potentially on a filter or through other means. Same thing goes for the air. But when we're taking a soil sample, it's, I don't know, you look pretty strong, Anja, but probably not taking up 1 ton of soil to then go search for microplastics inside, right? So I think that the sample size is much smaller, and also soil itself is made up of a lot of particles. So it's really finding a needle in a haystack. So we have questions about, when we take this small sample size, how representative is it of the entire fields that we're sampling? And also, how do we effectively isolate microplastics or nanoplastics from the soil in order to be able to quantify and characterize them? So in this case, I think that soils are oftentimes more challenging than some of the other matrices. And that's also likely why there are more studies looking at water than in soil currently.
Our knowledge on nanoplastics is increasing, and we still have open questions, but are there any conclusions that you can draw from your research already? Like, what's important to consider when we look for solutions to plastic pollution from your perspective?
So while it's not necessarily stemming from the direct research that we do in the lab, what is important in a solutions-oriented approach is to ultimately consider the entire life cycle of plastics, not just the pollution, and also not just nanoplastics, because most microplastics and nanoplastics are formed from other types of plastics materials that end up being mismanaged waste. So in terms of considering this entire life cycle, this starts already at the production of plastics. If we reduce the amount of production of primary plastics, then we have already taken one big step forward, not only because of the mismanaged plastic leakage that could get into the environment, but also because there are a lot of other environmentally unfriendly practices that go into the production of plastic itself. This can start already with the oil extraction or the petrol extraction, and also other parts of the production chain. When we think about where and how plastics are produced, this consumes quite a lot of energy, and energy production can also release a lot of greenhouse gas emissions. When we think about the design of the material, it's also an opportunity of where we can optimize the design of the item, either to have less plastic in a given item or to make it out of a different type of material if that's possible and suits the needs. And then we can also think about how to deal with the end of life of plastic. Now, waste management is not always ideal, and maybe in some Western areas, it's getting better and better, having more recycling, having better garbage collection. But in a lot of places, this simply isn't possible yet. So being able to have a better waste infrastructure across the globe, since this is a global pollutant, which will also transport across the globe, is really key to making an effective solution to reducing the amount of environmental and health impacts about plastics.
Thanks so much, Denise, for taking the time to share your insights and knowledge with us. I am very curious what you're up to next. Can you share anything about that yet?
Yeah, so one line of research that I think is really exciting for me personally is not only looking at the direct effects of microplastics, but also the indirect effects of microplastics and nanoplastics. So what I mean by this is not only taking a micro or nanoplastic-centric view, to be taking a sample and quantify how many plastics are there, but taking an environmental systems view to actually understand what is really changing in the environment with the presence of micro and nanoplastics. Two examples of this, one from our research in water and one from our research in soil. In water, when we think about how microplastics aggregating with other natural particles, we've worked on this case of plastic aggregation with algae. Now, algae naturally in a water body will be growing at the water surface, die, and then sink to the bottom. And this is part of the biological pump, which is important for carbon and nitrogen cycling. Now, we investigated when plastics are also present, does this expedite or not this settling and natural cycling of nutrients in water systems? In the context of soil, we're also looking at not just how plastic particles are moving through soil, but how it's actually changing the soil properties themselves. So when you have more or less small pore sizes, does this change the oxygen diffusivity through the soil? So ultimately changing the respiration of soil. Does it change how other nutrients can leach through soil when there's plastic particles are present? And does this depend on the plastic size, the plastic chemistry, or the soil properties themselves? So again, it's looking at what additional changes there are to environmental systems because plastics are there, not just the question of if plastics are there or how are they moving.
Great. Thanks so much, Denise, for being a guest on the podcast.
Thanks. It was wicked fun.